Murali Krishna Kumar, Associate Professor, Department of Pharmaceutical Sciences, Andhra University. I request Navya to come forward and give a brief intro about story. Now, very much great Kumar. It is absolute honor to introduce our esteemed speaker, Dr. Murali Krishna Kumar, for his distinguished academician, researcher, and thought leader in the field of pharmaceutical chemistry and drug discovery. With a career of spanning over 2 decades, Dr. Murali Krishna Kumar has made significant contributions to medicinal chemistry, computational drug design, and spectroscopy, shaping the landscape of pharmaceutical research and education. Dr. Murali Krishna holds PhD in pharmaceutical chemistry from Andhra University, where he has also completed his M.Com with outstanding academic credentials. His professional journey includes study as an associate professor at the College of Pharmaceutical Sciences, Andhra University, where he plays a pivotal role in research, mentorship, and academic leadership. Beyond academia, Dr. Murali Krishna has been at the forefront of innovative drug discovery, loading multiple research projects funded by prestigious agencies such as DSP, SERB, AICTE, and CSIR. NMITS. His research has focused on synthesis of novel bioactive compounds, tuberculosis drug development, and marine natural products, leading to groundbreaking discoveries. His work has also resulted in Indian patents in the universal derivatives for pharmaceutical applications. An accomplished researcher and scholar, Dr. Murali Krishna has authored over 60 research publications in reputed national and international journals. His expertise in animal spectroscopy, molecular modeling, and structural elucidation has positioned him as sought-after research person at conferences, seminars, and workshops. His contributions to academic research include delivering keynote lectures and sharing scientific sessions at prestigious pharmaceutical and biomedical forums. Dr. Murali Krishna's commitment to advancing pharmaceutical sciences extends to his administrative role, serving as the director of the NMR Research Center, placement officer and nodal officer for epilepsy, data management at Andhra University. His leadership and dedication have been instrumental in fostering academic excellence and individual collaboration. Students have been privileged hearing from a visionary pharmaceutical science whose research and expertise continue to drive innovation in drug discovery and therapy. Please join me. We welcome Dr. Murali Krishna Kumar, Brigadier General.
Hello.
Now, over to you, sir.
Yeah, can I present my talk?
Now you can share, sir.
Good morning to all of you. One second. Hello, Kiran, can you see the slides?
We can see the slides, sir, but your audio is little low, sir.
Okay, 1 second. Hello. Yeah, now is it okay?
Yes, sir. Now,
so good morning to all of you. First of all, I thank the organizers for giving me an opportunity to share my experience with all of you. So the topic that is given to me is the role of bioinformatics, mostly with a view to drug discovery. So in Andhra University, we were doing some research work to find new drug targets and also find new drugs or lead molecules in the academic environment with the available resources. We are designing methodologies to see that attrition, success rate is increased and time is saved and with low resources. So one of the topic where we got very good results is I am presenting before you as a case study, so that the young faculty over there may pick up some point over here, so that in the academic environment with the available resources within the college, they can do productive research work. So here my topic is host pathogen genome comparison, identification of possible drug targets. So that is one of the uh, project work that is going on in our laboratory for almost last 4 or 5 years. Yeah. So our research group is mainly focused on discovering drugs or drug targets for tuberculosis. We are working on this topic for the last 15 years. Mycobacterium tuberculosis, everybody knows that that is one of the oldest disease that we have. But the challenges that we have over here is like development of drug resistance, means the microbe is becoming so intelligent that it is using all its mechanisms to escape. Host protective mechanisms means the microbe enters into your body and stay there for long period of time without getting affected. Second one is developing drug resistance, means whenever you use a drug, it has such a flexible mechanism inside its biochemical apparatus to adapt, to change to the new, to face the new challenges. So drug resistance is coming. So that is forcing research teams to discover new drugs. As we can see, more and more resilient microbe needs a little bit more toxic drug. So that's why first line and second line, if you see, second line anti-TB drugs are rather toxic. Ternary is even more toxic. And here patient non-compliance is again another big problem we have to handle. Means the drugs that are given to them during the treatment, maybe because of the treatment period or because of doses or because of the toxic effects, patient non-compliance is an issue. So this is actually the background on which we are working. So one of the point that we pursued is microbe, when it is infecting our body, it should be detected by our immune system and it should be cleared from our body. But in case of Mycobacterium tuberculosis, what exactly happening is it is able to enter into your body, stay in the tissue, stay in your bone marrow, stay in your macrophage, still alive. It is adapting to the new environment and able to stay in the body for longer periods of time. So that is one point we were actually thinking of, and we are hypothesizing like this. What exactly may be the reason why Mycobacterium tuberculosis is able to stay in the host body for such a long period of time without getting detected? Maybe it is adopting its changing itself by monitoring host chemical signals. We are all aware that Mycobacterium tuberculosis or tuberculosis usually affects those people with a compromised immune system. Compromised immune system. So how come bacteria know that the person has more immunity or less immunity? Maybe it is actually monitoring some of the chemical messengers which are indicative of host immune system. That is one of the hypotheses we are actually pursuing. Second one is quantitative, means monitor the chemical signal levels. Maybe, excuse me. Maybe it is not only monitoring but also quantitatively analyzing. Third one is it is able to tune itself by altering its own biochemical pathways in response to host signals. Third one is it is changing its biochemical apparatus, like maybe enzymes are mutated or it is adopting by no through mutation in one or the other way. New type of proteins it may be preparing to in response to host signals. Fifth one is really dangerous. That is manipulating host biochemistry by supplying in situ produced host signals. These are the 5 points which we were actually pursuing. In the literature survey, what we found is the fifth point, manipulating host biochemistry by supplying in situ host signals. So especially here, we found solid evidence that Mycobacterium tuberculosis is preparing cyclic AMP and releasing it, especially in case of macrophage, so that the macrophage will lose its capacity to digest the bacteria. Because in case of macrophage, what will happen? It is engulfing the pathogen and then it will digest it by switching on enzymes and proton pump. So these two are actually manipulated by using by Mycobacterium tuberculosis. That was proven. But the remaining 4 parts are actually a little ambiguous because it results in identifying proteins which are like chimeric proteins. So here our journey is in brief. I'll try to tell in the next coming slides. So here we found one more interesting theme here, like our research group is also working on drug repurposing or drug repositioning, means using old drugs for new purposes. Because in case of tuberculosis, one of the major issue faced by the pharma industry is drug discovery is a highly expensive methodology, because they may spend hundreds of millions of dollars, and ultimately after discovery of drug they need some revenue out of it. But in case of tuberculosis, the moment you discover and declare a drug as a tuberculosis drug, that drug will go into regulatory authorities like the respective governments. So the private companies cannot have a say as far as sale of the drug is concerned. Very limited market will be there. That is the issue. Because an antibiotic, if it is declared as anti-TB, that antibiotic itself cannot be used for other purposes, other infections. That is the issue. And clinical trials, etc., all of them are really troublesome. So here we found like drug repurposing may be one of the very important concept useful in discovering affordable anti-TB medicines. So here these two are the two tracks which will merge in the coming slides. Don't get confused. So here, so it is more like this. When a new drug is discovered from old drug, like aspirin is there, when aspirin is actually discovered as an analgesic drug, but right now it is used as blood thinning agent. Okay, it's a side effect for aspirin. So side effects are new mechanism of actions. So here, diphenhydramine, an antihistaminic drug, is actually the lead molecule for the discovery of fluoxetine, an antidepressant drug. Antihistamine drug gave us antidepressant drug. So this is again another very important objective. So this is actually the mixing of the earlier slides. We found that in one of the report, thioridazine, an antipsychotic drug, was found to have potent anti-TB activity. We are all aware that thioridazine is an antipsychotic drug belonging to the category phenothiazines. So in case of phenothiazines, when your research team, let us say if you are trying for antipsychotic drug, you will not focus more and more on antibacterial activity. Maybe once in a while, randomly, you may screen your compound for antibacterial activity because high throughput screening is a possibility there. But you will not definitely go for anti-TB activity. So it is an accidental discovery. this actually caught our attention because thyroidazine is an antipsychotic drug. It actually inhibits or blocks dopamine D2 receptors. What is the meaning of that? It is actually occupying the receptor pocket that is meant for dopamine. So how come this dopamine D2 receptor blocker is inhibiting another completely different genera, that is Mycobacterium tuberculosis? So is there anything to do with the mechanism of action? Because for thyroidism, so far, no mechanism of action is proposed. They only said it actually alters the activity of lipid biosynthesis as well as mitochondrial activity. These are the only two reasons proposed, but the detailed mechanism is not yet known. So this actually enthused us. So we did some computational work and found through shape-based virtual screening. So we found other CNS drugs like fluoxetine, citalopram, sertraline, and many other drugs were also found to have anti-TB activity. They are all non-related. One of them is a serotonin reuptake inhibitor, another one is antihistaminic, another one is a dopamine blocker. There is no commonality in all of them. So this actually made us think maybe microbe is adapting to the host, or because Mycobacterium tuberculosis is a soil-borne microbe, one of the oldest as far as its evolution is concerned. So over a period of time, maybe it is closely interacting with the higher animals so that it is changing its biochemistry slowly. That is one of the reason why most probably Mycobacterium tuberculosis became so resilient. Once it enters into your body, it will not leave. It can hide anywhere, like from bone marrow to adipose tissue to your lungs to your CNS tissues. It has such a knack. Okay, so maybe because of this adaptation mechanisms. So here, this is one of the reason. This could be one of the reason. So it may be monitoring one of our chemical signaling mechanisms. So if it is able to do that, let us further the hypothesis. So let us say if Mycobacterium tuberculosis is monitoring our signals. For example, serotonin. If it is monitoring our serotonin levels, then it should be having a receptor. It should be having a receptor with serotonin receptor site, a protein with serotonin receptor site. Maybe it is similar or not similar to our actual protein. That is the second line of discussion. But it should be having a receptor where it accepts serotonin. So that is what is our hypothesis. So we wanted to test this. How to do this? So we found this strategy. This is our plan of work. So what we really wanted to do is take the uh, the receptor protein or gene of host, that is for us human proteome or human genome. You take any one of them, and let us say we are trying to find out if serotonin receptors are there or not, 5-HT receptors. Then take all the 5-HT receptor genes and search in the genome of Mycobacterium tuberculosis. Fortunately, Mycobacterium tuberculosis whole genome is now available online. So that is the attempt we made. In the beginning, we thought like it is an easy task, searching for something in Google. But later on, we realized so many complicated problems. So we started with all these signaling molecules, starting from histamine to adrenaline, noradrenaline, serotonin, dopamine, and others also. And then we also tried the channels. So we do have channels, okay, sodium channel, potassium channel, etc. This is a huge project. So we have obtained several positive results. So one of the result I am presenting before you. So the plan of work is Like first we pick up the gene and do the blast analysis. And then for the hit molecule, whatever the hit molecule that we obtain, we go for homology modeling. And then we'll try to do the docking for validation of the protein. And then we will perform anti-tubercular activity, that is in vitro activity. So this is the way how we are actually planning. So here the target drug proteins. The target proteins means in humans, the proteins that are there, selective, because we cannot search for every protein. So we pick up one class of proteins and then search in mycobacterium protein proteome. And then when a hit is obtained, if modeling is needed, we go for homology modeling. Then we go for screening. This is the way. So here what we will probably obtain, let us say if we have a dopamine receptor and search in the mycobacterium tuberculosis, if we are able to find out or if a hit is obtained with a dopamine receptor containing protein, then it indicates that if you give any chemical which modifies dopamine receptor transmission in host, it may probably carry anti-TB activity. So that is our proposal. That is our proposal. Let us say if dopamine receptor is already there, containing protein, if it is there in host, in the pathogen, there is a possibility. So that is one of the reasonable rationale that we are proposing. So what we have done is we have taken so many. Here one of the exciting result is we have taken the ion channels. Ion channels. So here we have taken potassium channel, chloride channel, sodium potassium, ATPase. So all these. So here when we have taken sodium potassium ATPase of a human, the protein, and search in mycobacterium tuberculosis, then we found these 4 hits. So the hit percentage is 31.52, 98% query cover is there. Means the protein is uh, related protein is there in mycobacterium tuberculosis. Then because for that protein in the literature, in the database, it is mentioned as maybe it is a sodium potassium ATPase, sodium pump P type. And then for that, we don't have any 3D structure available. 3D structure available in the database, that is PDB database. So we have to do the 3D modeling also. So we picked up homology modeling because online resources are good enough. Swiss Modeler is one of the most dependable modeling environment available online, but that is limited to globular proteins of enzyme category. Most of the transmembrane proteins, for them, obtaining dependable model by using Swiss modeler is not that easy. And in our experience, we are not happy with Swiss modeler. So there we found Robeta is another modeling environment where we can use multiple templates parallelly to build a model. That means, let us say if we have 1,000 amino acid containing protein, if it is there in Swiss modeler, we can take one template or related templates for building the model. So in case of Robeta, let us say in 1,000 amino acids, first 200 amino acids are matching with one protein, 300 to 600 another protein, 700 to 800 another protein, and the remaining unknown. Even in that type of condition also, Robeta modeler can give you a very, very dependable, reasonable model. So we tried that. So here, this is the regular homology modeling protocol, query selection, then selection of MTB protein for homology modeling, a 3D structure of protein, and then model validation, and then active site prediction. All these are routine steps that we follow in case of homology modeling, especially in the drug discovery cycle. So this is the one of the results that we found. where the appreciable score of 31.52% is obtained. That means human protein and the mycobacterial protein, when we did that, 31% similarity was found. And the protein in the mycobacterium tuberculosis is also sodium potassium ATPase. And then homology modeling was done by using Roberta. Swiss modeler is not working actually, because when Roberta modeler was used, model validation is done by using standard SOP is there, like Ramachandran plot analysis and then Z-score value, verify 3D plot. All these can be done online by using the resources. And then we got appreciable validation results after the verification of the 3D structure of protein. So here is the final results. If we closely observe, this is the blue colored one is human sodium potassium ATPase, and the red color one is mycobacterium tuberculosis, the channel. When we compare these two, on the larger side, they appear like similar, because the number of folds, number of alpha helices, number of beta helices, their arrangement, certain loops are changing. In some areas, loops are missing, but overall, the alignment of the protein remained the same. For that, we were happy. But these two belong to two genera. They cannot be 100% homology between these two. So here, our objective is to see whether that sodium recognition points as well as the other the ligand reception points, whatever are there, if they are there, then we will be able to say if any use is there. For example, sodium potassium ATPase is actually the receptor for digoxin, cardiac glycosides. Cardiac glycosides actually act on this receptor. So that point we have taken to see whether digoxin reception amino acids in the receptor pocket, if they are there or not. Yeah, this is the comparison of active site amino acids for human sodium potassium ATPase and mycobacterium sodium potassium ATPase. All these red colored amino acids are very, very important, crucial as far as the reception of digoxin or sodium are concerned. We were happy to see that their alignment remained the same. For example, valine, alanine, asparagine, valine, valine, alanine, asparagine, valine. And then the alignment of the remaining, because the numbers may be changing because the size of the protein is different, but their alignment remained the same. So we thought like maybe we got a protein similar to human protein, and now we wanted to check whether our hypothesis is correct or not. So what is the molecule available for us for checking this? Nothing more, none other than digoxin only. So we wanted to verify in silico whether our hypothesis works or not. Then with the digoxin, we have done the docking and compared it with the existing results. We found very good score of -9.1 in autodock. And the way how it aligned at the receptor pocket actually matched with the human receptor. That gave us confidence to say, yes, we got a receptor pocket similar to host receptor. Then we wanted to verify if it still works. So we have taken digoxin from the market, we purchased it and performed anti-TB activity in our TB screening facility, anti-TB screening facility. Anti-TB activity of digoxin and digoxygenin, they were found to be 25 microgram per ml. Actually, sorry, there is a typo here, and 0.8 microgram per ml, because digoxin contains digitoxos, because the sugar unit is there, that can be hydrolyzed. Digoxygenin can be obtained by using simple hydrolytic procedure. Enzymatic hydrolysis is there. We got it from the market again. Digoxygenin showed even more activity. And after this, because 0.8 microgram per ml is equal to any standard anti-TB medication. Because if you see the rifampicin, its sensitivity in especially this microbe is approximately 3.25 to 1.6. In that range, we get the MIC value. And the digoxin, even in double-blind screening protocol also, we obtained 0.8 microgram per ml. That is actually very, very interesting, highly potent. So in this way, we actually made a hypothesis, used computational resources, a little bit of logic, and minimal resources that are available in our facility to obtain very good result. So to validate whether it really works, there is a biotechnological procedure we have to follow, molecular biology procedure, to say whether this receptor actually is the drug target. Can we explore this further as a drug target? Because digoxin is a cardiac drug. Can we use it as a drug? No, we cannot use it. But we know that QSR is there. The moment you open the lactone ring, the moment you break the glycosidic linkages, its potency will go down significantly. So that is the attempt we made. We broke open the hydrolyzed the lactone ring, and still we found very good biological activity for digoxin. That is what I did not put it here, but we have done it also. So that is the way how, in a reasonable period of time, by using minimal resources, we'll be able to discover new targets by using this strategy, especially for antimicrobial kind of drug discovery. So here what we have done, we have hypothesized something by taking into consideration existing results obtained in the drug repurposing studies. And then we tried to search for similar proteins, host-like proteins in the pathogen. We found the protein and did the modeling and validated the protein in silico as well as in vitro. So this is the way how we are able to find a very good potent anti-TB activity in cardiac glycosides. So that is really interesting. So thank you all for your patient listening. Now the session is open for discussion. I hope I made myself clear. I hope you enjoyed my lecture. Hello.
So thank you, sir. So we'll open the session for questions.
Yeah, sure. Hello.
Sir, yes, sir.
Good morning, sir.
Good morning.
Yeah, how much similarity you look between those two ATPases, sir, sodium potassium ATPases?
31% is there.
Is there any rule for the similarity, sir? Like,
yes, of course, because more the similarity, better the model.
okay,
because we can have 10%, 15% similarity also. Sometimes it may be useful to us, but it will not give us a dependable model.
If there is more similarity, then the drug will act both on the host and the pathogen, sir.
There is every possibility, yes.
So how you will try to reduce this action on the host, sir? Because it became a side effect, right?
Yes, you are right. But the thing is, the pathogen protein is not absolutely similar to host protein, and it will not follow the QSR that we have. Let us say, for example, in our case study, digoxin is working as anti-TB active. It has anti-TB activity. But the moment you open the lactone ring, it will lose its activity on the host. The moment we break open the lactone ring in cardiac glycosides, it will lose its biological activity. That is, cardiac activity is gone. But we found still potent anti-TB activity is there.
So you're telling we can make it a more specific towards the pathogen.
Yes, yes. Because for the existing drugs, fortunately, we have a lot of data available because side effects, QSAR, other data, success or failure data, everything is available. So we can make use of that.
Oh, nice work, sir. Thank you.
Yeah, thank you.
Any more questions we have? If any more questions we have, we'll take up. One more question. Now, we request here to propose thanks for. Yes, sir.
Thank you, sir. Thank you for your interactive session. I hope everyone got their insight from this.
So thank you, sir. Thank you for your
Thank
presence.
you. Thank you so much. Okay, I'm leaving. I'm signing.
పైన సిట్టింగ్ చేసినప్పుడు కోడ్ ఇంకోటి జరుగుతుంది.
జరుగుతుంది అలాగే